Pretty good. I'm going on vacation next week, so I'm busy because I'm trying to sort of get ready for.
Oh, yeah.
So anyway, so Monday morning, I'm probably. Well, I might be able to make the meeting next week. I'm not sure.
All right. Yeah, if you can. Yeah. Well, good luck with your vacation. You seem like you've been working very hard to deserve it.
Well, I don't know about hard, but sort of trying to find my way.
Well, yeah, that's a lot of work. It's just.
Yeah, they're asking for equivalent math, like he did finite element analysis and found the equation for the stiffness matrix, and so he said find the one for tensegrities.
Yeah.
And apparently it's called what? I don't know. It's called something different. I could go find it.
Yeah.
I should remember that.
Hello, Karan. How are you?
Hi, Brad.
Hi. How are you? What's going on these days with you?
I started reading up on two, three, articles related to how biology is kind of like the neurological origins of biology. Some books that were written by I think Robert Sapolsky. So like somebody suggested those to me. So I was kind of like this is a new field, like kind of going through stuff there. Other than that, even I think Dr. Gordon had given that hydrophobicity task, right? So I'm still trying to go through this weird branch of archaea and how they kind of behave and how they interact, their membranes, they kind of some tiny details. I think that was that.
Yeah.
Yeah.
Yeah, that sounds
And
good.
also related to the axolotl plot data, right? I not really like I'm getting somewhere, but I've not really reached a particular place so far. With the especially with the image data sets, right, of axolotl egg images in advanced stages. So when it comes to grouping those clusters and trying to formulate a 3D image of that sense,
It's probably difficult because they're at different angles. Like
yeah.
it's flipping, so the egg doesn't follow the flipping.
Yeah, I at least be able to get a rough estimate of that 3D structure. So that won't take that much time, but for the time being, I'm just trying to increase and decrease the contrast values in such a way that I can at least give it much more better contrast. Like these are the contrasting images that I can see right now, the neural net. So
I really need to get some new image.
Yeah, okay.
So that
I'll try to work with these as well for the time being. Otherwise, I think because how do I explain this? Maybe with respect to how those group of clusters are labeled at they quite like they're three images in sync with each other, right? They're like slightly after a very slight angle variation. That portion is visible and the backward portion is visible in like three very no, was it this? I think because of the column that is also present and the which let me open my Google Drive and try to explain it from that.
Okay.
I've also run into photogrammetry. Photogrammetry might help give a 3D image of of the salamander eggs if you've got all sides of it. And like I said, the new microscope probably is what I need to use to get some new images, because the angle doesn't change. The angle of the images doesn't change. So I think it's going to be better. But so far, the salamanders have decided not to lay eggs. So it was a bad year for them here because it was dry and the water quality went down, and all of our salamanders suffered. This is Elizabeth's and mine. Elizabeth usually had better water and better salamanders, but they're all not laying eggs. So we're working on it slowly.
Okay. I mean, at least I think by at least 2 or 3 weeks, I'll at least get the rough pictures down. Like at least have some general 3D structure of the axolotl egg in different stages. Like that won't take that much time. It's still the refinement part. The more I refine those images, the better data structure, that 3D model I'll be able to create.
Okay.
So it's just still kind of going through things that I can do with the current data set.
Yeah, you're working with the tough images first.
Yeah.
Yeah, so we'll have to give them pep pills or something, the salamanders. I don't know what to do for them. And by the way, my finite element analysis and the stiffness matrix, I found what it's called. It's called the flexibility matrix. When it's in the Tensegrity. If anybody wants to know.
Yeah.
Yeah. I did find it, but it's still confusing me slightly.
Yeah. Hello, Dick. How are you?
Okay.
Good.
Okay, I just came in. I missed what Susan was talking about, but
I was shown finite element analysis last week by Dr. Zhang, and Dr. Sharif said, find the one for Tensegrity that's exactly like that. It's not the same thing, really. Related.
okay. Well, in regard to the axolotls, they can be finicky. We used to drive them around the city, and then they would mate.
Yeah.
The puppy ride was inspiring.
I maybe should tell Elizabeth that. Yeah.
All right. So, is there anything we had to follow up from last week? I know we talked about Tensegrity and we talked about some of the other talks, image processing talks, and that. Did we have any questions or?
No, I still have this. See?
Oh, there we go. That's Tensegrity
toy.
toy.
A couple of more.
Yeah, mine doesn't work anymore. The plastic legs.
You can replace the plastic with just string and it still works.
Oh. Okay.
Yeah.
So I had a question about that toy, or like in general, a Tensegrity structure. So it's the end; those strings at the ends, or they're pulling and pushing, or they're exhibiting all like opposite forces, or how exactly is it being held together?
They're usually elastic.
Okay. The elastics are in tension. That's why they're called a Tensegrity structure. They are already in tension, and they're pulling, and the compression elements are the rods.
Okay, and so
this cell is a microtubule.
Okay.
Right, Dick? He's the one that told me this.
Yeah, the analogy would be that the microtubules presumably are much stiffer than microfilaments, and the microfilaments are contractile. Okay. So the microfilaments would be conceptually represented by the elastic components. Problem is, on the real Tensegrity toy, the elastic components decompose after 20 years.
Well, they make them with string. Like, you don't have to have an elastic in there.
Same may happen to a string though.
Oh, yeah. It's not active matter. It's not reproducing itself or whatever
That's
the cell does.
not self-restore.
Yeah.
Yeah.
Good. I just thought that might be useful to clarify what that was or how that worked and how that sort of holds together. So I may not.
There's YouTube videos about this. People building.
Yeah.
But none with motor molecules attached.
No.
No, well,
Or what they call dynamic instability, which means that the elements kept lengthening and shortening continuously.
yeah, they don't have. It's not an active matter.
Yes.
They haven't built an active matter one.
That's what deserves simulation, to actually have a tensegrity structure that's dynamic and
Yeah, you can add energy to it.
yes.
Have it do something.
Yes.
Oops, we lost Mainak. Where did he go?
Oh, he probably just had to reconnect.
Okay.
There he is.
Okay.
Oh, my knock.
Hello?
There you are. Yeah, I can hear you, I think. Oh,
Yeah, I
Okay,
lost
there.
connection for a moment there.
oh okay. So, how are you doing?
Oh, I'm doing just fine. I'm doing
Good.
great.
Good. Okay. So that's, let's see. Oh, Richard, Dick asked Karan, interested in mixed chirality problem. I guess that's something you discussed before, or?
Yeah.
Okay.
I think last one.
I'll turn off my bandwidth here.
Okay.
Yeah. Hello.
Hello.
Hi. Hi, Doctor. Yeah. Is this a different problem, or is it the same one?
Yeah, same. The problem predicting the hydrophobicity
Yeah.
of a mixed chirality peptide, right? I doubt that's in the literature. Probably a new problem.
Okay.
Okay. Oh.
Hi, Max. Go on again.
Okay. Yeah.
Oh, okay.
So yeah, I don't know if there was anything more to say about that or.
Well, I was going to do a very crude simulation, and then I realized the problem is very complicated.
Oh yeah. Yeah.
The simulation software that I'm getting for free, because it's not free. It's through the university. It's called ComSol, and it has a chemistry section, or
Yeah.
has
Well, if you make a peptide around 20 amino acid residues, and they're random selection in terms of whether they're L or D amino acids, and you calculate the hydrophobicity, that's basically the problem. Okay?
hydrophobicity.
Yeah, and they're not from racemic mixtures, because the preponderance of evidence seems to be that the amino acids from meteorites, which are presumed to be the ones at the origin of life, are biased towards L compared to D amino acids, but it's an incomplete bias. So that can be included in a simulation, so you get more L than D.
Okay, so biased.
Okay? And then there are other weird phenomena, like once you start an L alpha helix, it seems to be biased, so it will propagate its own kind. And there are some subtle differences between L and D, which might also introduce some bias. So it's an interesting problem. And then there's also glycine, which is not chiral at all, which is also the predominant amino acid for meteorites. Okay. So I've worked up tables of estimates of the amount of LMD for the various amino acids.
Huh. Okay, and nobody's done any work on that?
Nope. Not that I can find.
Um,
The problem is nasty because even if you only confine yourself to L amino acids, the prediction compared to experiments are often wrong. Okay, so even where we think we know what's going on, nobody's done it right yet.
huh. Now I'm even busier. I don't know if I can do that with the console, but it's
This
not.
takes, this takes really detailed molecular dynamics.
Okay, they're
Probably.
building structures with DNA.
Oh yeah, that's fine. DNA is all L.
It's all L. DNA
Yeah.
is L? Really?
Yeah, there's an L and D version, I think, of nucleotides also. You can check that out.
But you're talking RNA here?
No, we're talking peptide.
Just peptides. Oh, okay. Yeah, peptides.
Peptides in membranes, and for now I'm considering the membrane as not chiral itself, which is also an approximation. Okay, because at least membranes that exist now are chiral. The lipids, or the phospholipids, that can be chiral.
They're all chiral?
No, many are chiral. I don't know how many. I haven't done a survey of them.
Are probably chiral.
But the initial ones probably, well, maybe we're not. Okay, it's hard to know.
Okay. Well, that should, yeah, definitely that's a part of how things work. So,
Yes. So it's subtle.
yeah.
Okay. Yeah, sounds like an interesting problem. So I'm gonna share my screen for a minute. I think we talked about a couple weeks ago. We talked about Ising models, and I don't know where we are on that, but
We're in a state of inertia.
okay, yeah. So this is the Wikipedia page in Ising models. I don't know. We missed out on it last time showing it. I just wanted to show it real quickly.
Okay. The site's too small to see here.
Okay, so this is, let's see if I can zoom in here a bit more. I don't know if that's bigger.
Well, it's a little bit bigger.
Yeah,
It
yeah.
needs to be about 2 or 3 times bigger.
Okay.
Maybe my eyes are going.
Well, yeah. Sometimes it doesn't project at full resolution through the, but, so this is the Ising model, named after physicists Ernst Ising and Wilhelm Lenz, is a mathematical model of ferromagnetism and statistical mechanics. So this is something that, people have developed models in statistical mechanics. We often borrow them for biology, especially if it's biophysics, but also people use it for more ambitious, types of systems like, traffic systems and things like that. People will use statistical mechanics sort of as the basis for their model. So there's a lot of good mathematics in statistical mechanics. This is one sort of model in that area. And so this particular model, it consists of discrete variables that represent magnetic dipole moments of atomic spins. So you're looking at things that have these spins, and they can be one of 2 states. So this is +1 and -1. So this is very similar to a binary model in a computer science or a computer science model of a bit, where we have 2 states, +1 or, we usually have 0 or +1. This is +1 or -1, and it's usually the polarity. These spins are arranged in a graph, usually a lattice, and then allowing each spin to interact with its neighbors. And then the neighboring spins that agree have a lower energy than those that disagree. So they minimize their energy if they agree. The system tends to the lowest energy, but it disturbs this tendency. So you can apply heat, some analog of heat, or you can have this sort of agreement between the states, and this gives you this lowest energy. And so you look at the lowest energy states. So this is basically the approach, and so they kind of go through a lot of the mathematics of it. So this is, so let's see if there's anything. I guess there are connections to graph theory, which I found interesting. And there are other connections, there may be connections to machine learning here as well, especially with respect to energy minimization. I know there are some people who are doing I've seen some papers where people are kind of starting to make the connection between like energy minimization and statistical mechanics and like gradient descent in machine learning. Basically, the gradient descent is this minimization of state, minimization of energy, as they define it in, say, a machine learning model of error, and then there's this minimization of energy as well in statistical mechanics. And like I said last week, we talked about fitness landscapes. That's a very similar model where you have this, in this case, you might be maximizing fitness. And so it's just flipping that over and saying, what's the maximal fitness? So, you're looking at a bunch of states, which are these individuals in a population. And the population, not the entire population, is maximizing its fitness. And then that's your distribution of states, whether they're maximized or not. And so it's a very similar sort of thing across all those areas. People are using those kind of similar, they're using, mathematical models, but they're also using this broader set of analogies. And so I just wanted to point that out for people, just maybe to get people interested in some of these topics and then seeing what the connections are. I know we didn't have a lot of time last week to talk about the connections between, different areas. I mentioned it briefly, but then they have this thing about Monte Carlo methods. So they use this, quenching approach where they heat the system up and then they cool it off. So it's quenching. You can use that as well. People use that in simulated annealing, which is a computer science technique, which is somewhat similar to this, where you apply heat, which is basically you basically apply noise or randomization to the problem, and then you let the problem cool and then see what settles down to the minima, and then you select that. So there is that sort of analogy between between statistical mechanics and physics, and then some of the computer science models. So I mean, I don't know. Just wanted to point that out. It's just something I found interesting.
Okay.
This week.
Bradley, can I give some historical context here?
Yeah.
Ising came up with that around 1930 in a one-dimensional lattice. Okay?
Yeah.
This is, of course, before computers. He presumed that would represent a phase transition for ferromagnetism. And he was wrong. Okay? It did not produce a clean, sharp phase transition. Then later, somebody discovered that if you make it two-dimensional, you do get a sharp phase transition. And then the excitement went on. Because it became the first simple model for a phase transition of the material. Okay, now, it's also applicable to what's called a lattice gas. A lattice gas means simply that zeros and ones represent empty and occupied of some molecule on some lattice. Same kind of interactions between neighbors. Okay, so I got involved with this in the 1960s in terms of lattice gases, and that's where my interest comes from. Okay, now if you go back and take the one-dimensional model and make it two-dimensional by making the other dimension the states versus time, then you get something akin to a Wolfram pattern. And this has raised questions like, what's the relationship between such a propagated one-dimensional Ising lattice gas model and Wolfram patterns, especially the ones that are claimed to be chaotic. Okay?
Okay.
And the other question which came up, which was kind of interesting, that is, if we generalize the Wolfram patterns in any way, are they closed? In other words, do you generate any new patterns, or can all new patterns that you get be classified under Wolfram's 256 rules, sets of rules? Okay?
Yeah.
So there's a curious mathematical problem there.
Yeah.
Closure. Okay.
Yeah.
Now I suspect they may not be closed, because if you take a standard Wolfram pattern, and for each pixel in the Wolfram pattern you put a picture, okay, then the pattern will be a pattern of pictures. Okay, this is sort of a generalization of the idea of making a picture of pictures, where each pixel is replaced by a picture. Okay? And I think in some sense that's not closed. I'm not sure. So it takes a careful analysis of what the definition of closure means in this context.
All right. Yeah.
Okay, you're gonna have to explain that more to me later.
Okay, closure, take any two integers. If you add them, you get an integer.
Actually, okay, no, I'm sort of know about closure, but I don't know about Wolfram patterns.
Oh, you don't know about Wolfram patterns? I don't know. Bradley, you got one handy?
I don't think so, but
Just
let's see
look
if I
up
can.
Wolfram
Yeah.
pattern in a search for images.
Okay,
Let's see. Rule 110 might be useful. Yeah. So something like this, maybe. So this is. Oh, I gotta share my screen first. Yeah. Okay. So I think like something like this is a Wolfram. This is rule 110, and this is something that's generated by this simple rule, and it's just applied recursively. And it's like basically forming your triangle, and then you just keep applying this, and you get this edge here where you have larger triangles, and you go down to finer and finer scales.
Yeah, basically, see, if you take the horizontal line at the beginning, that's how it starts. Mostly zeros and maybe a couple of ones. And then you apply a set of rules. There are 256 sets of rules. You pick one of them and you just propagate that rule deterministically, and this is the pattern you get. Each horizontal line being a time step. As you go down the page, time is 0 at the top and goes down.
Yeah.
Okay, now the biological interest in this has mostly been that some of the patterns look like some of the snail patterns.
Right.
But nobody's ever tested that quantitatively.
Yeah. It's also self-replication too, that you have this process that. Right.
It's related to the game of life and propagating patterns and whatnot.
Right.
Self-reproducing patterns.
Right. I've always found interesting that's a like I've seen the people some of the papers on that, and it seems like it's a little too abstract to be useful. Like there are ways maybe you could build trees from this that are like really informative.
Yeah, well, yeah, yes and no. Wolfram himself made this the basis of his new physics.
Right.
Where he's going to replace all physics by this.
Yeah. But yeah.
Which also then the question of closure is very important.
Right.
Because physics touts itself on being predictable. And if you change the rules different from the ones Wolfram used, is it still closed?
Right.
Hello, Jesse.
Oh, hello, Jesse. How are you? You have something in the chat? Oh, okay. Jesse says, hi, all. Won't be able to speak much, but just checking in. So, hello. We haven't seen him since the new year, so. It looks like Mainak is back as well, so good. So, yeah, we were talking about these sorts of things. So, if people are interested in this topic, by all means, we can discuss this further. Maybe someone can present it. Someone wants to do a little simulation.
The problem is the literature is huge. I did a survey of one-dimensionalizing models and came up with 800 papers.
Yeah.
Okay, so clearly we need to find reviews. We need to do sampling. Not all 800.
Yeah.
Okay, and we might want to make up some rules nobody's looked at.
Yeah.
See, all of the rules in the Wolfram patterns are nearest neighbor, but you could add second neighbor, things like that.
Right. And so it would give you a larger set of rules, perhaps.
Perhaps, yeah. Yeah, I think in fact there are some rules which give clean phase transitions for Ising models.
Okay.
Okay. Even though the original Ising, one-dimensional Ising model did not give a clean fight. You know what a phase transition is? Look
Well,
it's like water freezing to ice. There's nothing in between.
Yeah. Yeah, it's the first order phase transition.
Yeah,
So
first
it goes
order phase transition.
right from one state to another.
Yeah, yeah. And the regularizing model, if you make it two-dimensional, it does give you a first order phase transition. And I'm not counting literature on that. That probably goes to tens of thousands.
Yeah.
Called sublimation then. Is that second order?
I think so.
Sublimation? I don't know.
So well.
Sublimation would be analogous to absorption, desorption onto a surface. And I simulated that a long time ago, but I think that was first order.
Oh, okay. And by the way, what is Rule 30?
What is Rule 30?
It's interesting here.
Let's
So,
see.
what is it?
I actually had 110. I think I can go back to 30 here. So rule 30 is this. Go back to the search here. Why is it doing that? Okay, there we go. So rule 30 is this. It's a snail shell. So this is the Kona shell, which is similar to rule 30. You can see the triangles in there. And then down here you can see a pattern here on the lattice. Yeah.
Yeah. And most people have not gotten beyond the analogy stage.
Right.
It's sort of like saying that cracked mud is a good model for the patterns on the backs of turtles.
Yeah.
Because they look the same.
Right. So yeah, and then I guess there's some rule for chaos here. So chaos is another huge area.
I think Susan's question is, can you tell her exactly what goes on in each step? In other words, what does the rule look like and how is it applied?
So I think this is the rule set here. In all of Wolfram's elementary cellular automata, an infinite one-dimensional array, only two states is considered. Then this is the sort of the truth table for the different patterns. So for current pattern 111, you get a new state 0. So I think it's like you get these rule sets. So the next state from this previous state is generated by this truth table. You have like
the truth table includes what, the left and right neighbors? Does it include the one before?
I think so. Like this middle one, maybe. They don't really break this out, but I think it does include the one previous to that. So
Does it include the state of the pixel itself, or just?
New state for center cell is the one that's generated. So no, that's
Center cell. So it's 1,
Yeah.
2, 3, 4. So it's 2 to the fourth states, which are applied. And how do you pick? What's the rule? Whether you go 0 or 1, right?
yeah. So each of these string of 3 binary numbers generates a new state. So if you have a 1 and I guess it would be like 2 1s in the state, it generates a 0. If you have 3 zeros, it generates a 0. I don't know if that's a rule, but basically it's generating these different. So this is basically like 0 through 7 if you think about this in decimal. So this is 0, this is 1, 2, 3, 4, 5, 6, 7. And these are just for the different cells surrounding the center cell. So this could be in time. I think this is like a time step previous, and then it generates a new state in this center cell. So you'd have like in the neighborhood here, you have I think you basically how many of these either it's like the state of the different cells around it, or how many of them are of state 1. I'm not really sure how they count this, but so the corresponding formula is left cell exclusive or central cell or right cell. So that's the rule. In binary, you have this 0, 0, 0, 1, 1, 1, 1, 0, 2, and then 30. So it equals 30. So it's basically they're using binary to calculate the that's what 30 is supposed to mean. So I don't know. I might do a deeper dive into this. It might be useful to find interest, because there are a lot of different rules, and just to kind of say what is this? What would be interesting here is if we could test it out on different things, not just these simple patterns, but think about can we create a case where you have be like a 3-dimensional cellular automata, or what are the consequences of these rules? I don't know if people are into doing some sort of mathematical analysis of this, but that might be something interesting if we could just explore this and pull it apart and see what it's doing. Because I'm not entirely clear on what it's doing, but it might be useful because we had this paper a couple weeks ago where they talked about seashell patterns, which are these things up here. And they talked about how they're generated by some neural mechanism. So there's this secretion system that's based on some set of neurons in the snail, and they generate this pattern with this secretion system. But of course, it has to be put there in a way that's, based on some sort of pattern formation. And so that's maybe where Rule 30 comes in. Or as Dick says, maybe Rule 30 doesn't have a lot to do with it. Maybe it's just that it looks very similar to it. So we don't really know.
Yeah,
exactly.
It's also a question of what you ignore. If you go to the snail.
Yeah. Up here.
Put the snail picture up again. Yeah. The dark brown splotches are not part of the presumed simulation.
Right. Yeah.
Okay. And yet they seem roughly to form a pattern themselves.
Yeah. And so there are things like environmental noise, for example, that might play a role in this. And so if we made oh, go ahead.
It's also could be selective vision.
Yeah, it could be.
We see what we want to see. We only see Wolfram patterns because that's what we know.
Yeah. It's possible. I mean, yeah. So that's I don't know if anyone has anything else to say on that, or you have something in the chat. Okay, Mainak says Rule 30 is super interesting. Interesting video where Stephen Wolfram talks about Rule 30 and what makes it special. So this YouTube video in the chat is something that you might want to check out.
Okay.
So, yeah, if people want to follow up on that, please let me know. I might follow up on it myself, actually. We'll see. It might lead to something interesting. So, I'm going to switch gears, and maybe it isn't switching gears so much, but I'm going to talk a little bit about an abstract that I want to submit to NetSci. So, we have this conference every year called NetSci, and we had it last year. I don't think I have a link to it in here. But this is something that's being held virtually again. And I've been working on this idea for a while. So this is C. elegans. This is the embryo C. elegans. It goes from this is an animated GIF. Okay, it starts out in a couple cells. And then the cells start dividing and differentiating. And then you end up with this comma, and then you end up with this worm that's coming up. It's a little too fast to really follow it, but you can see that it starts from a couple cells, they divide, and then they form this comma. It's kind of it's changing its shape, and then it's twisting around like a pretzel, and then it's forming this worm here, which hatches out of the egg, and then it becomes a larval worm. So this is what's happening in the C. elegans embryo. This all unfolds according to this lineage tree. So we have this lineage tree that's what they call deterministic. So there are sublineages that have those cells are going to eventually differentiate and have a specific function in this embryo as we move towards this twisted worm. And when we get down to the bottom, when we get to these differentiated cells, they go from like a couple of cells in the egg here to a bunch of cells that are moved around in space. So you have to go from this lineage tree where there's a basic polarity of the cells, and then that switches around a couple times because the cells are moving around with respect to the anatomy, and then they shift around. So this AB is like at the front of the worm at the anterior end. The P1 is at the posterior end. But these A, B cells actually form all sorts of tissue type cells that represent tissue types, and they tend to be in different parts of the worm. So they have to move around. So you see there's a lot of switching in this tree from this straight lineage to these different places. So there's this process of sorting going on in the anatomy. So one way to deal with that is to look at, and I mentioned this before, we have the embryo networks where we have all the cells that we can look at use like the centroids as markers or something else, and we can build a network, a proximity network that mimics perhaps cell signaling or other types of substructure within this embryo. So you have like different cells that are forming, and as they're moving around and as they're forming tissues, they have maybe exhibit different motifs in that network structure. So this is a tree of heredity, but what I'm talking about is a network of proximity more than heredity. So if you combine those two, what's that going to look like? And so this example here is just from it's just a drawing that people have made based on the relative position in the adult worm, and then this hereditary unfolding here. And so there's it's not quantitative at all, really. I mean, it's just kind of a drawing. So I've been working on this idea of using a hypergraph to characterize this, or what I call sometimes a hyperlineage tree. And so I think I presented this before. I don't know if I presented I think a drawing of it, but I worked it out a little bit here. So this is basically a computational model of this process. So you have this tree where you start from a single cell, you move to 2 cells, to 4 cells, and then at the 4th cell you start to get differentiation by function. You get 7 cells that are in this 8-cell embryo that are somatic cells, and then you get 1 cell which is a germ cell, and then you follow this out down this germ line and you start to get more and more germ cells. These will become the eggs that are carried around in the egg sac of the worm, and all hermaphrodites have a sac of eggs. So this is actually quite common in the C. elegans anatomy, but this is separated from the somatic cells. Now, in this lineage here, you have these somatic cells that divide and they form these networks. So each of these circles is basically a network. In the case of one cell, there's no network; it's just the cell. And a 2-cell, it's trivial. 4-cell is kind of trivial as well. And then you start to get to like 12 and 24 cells when you get maybe like networks that are interesting. But in the germline, we don't really think that there's any sort of reason to have, well, maybe there is a reason to think that proximity plays a role in the germline. We don't really know. But the idea here is that you have these colored circles that represent different like sort of parts of or subgraphs of this main graph. So this embryo is going, as it differentiates, is going to have like a full graph of proximity and then subgraphs. So when it gets to this comma stage, for example, this tail end is going to have a subgraph. This other part is going to have a subgraph. Or as you start to get tissues forming, there are areas of the anatomy that might have subgraphs, like muscle area, where you have muscle, you have the connectome in the brain, and these sorts of things. So those are all kind of arranged in subgraphs. And so in this case, well, this is different. In this case, you have these subgraphs. So these white circles are the connectome, the neural connectome. These orange patches are embryo networks. So this is like all the cells that are somatic cells that aren't neural connectome. And then these yellow circles are the germline, and they all have their own subgraphs, and they all connect to one another. So what's interesting is that as differentiation proceeds, these cells in the embryo network, the reason I don't say somatic is because some of them are not differentiated yet. And so you have this subgraph here where you have exchange across to different subgraphs. So for example, here you have an embryo network of 194 cells, or you have 194 cells in this subgraph, 50 in this subgraph. For the next step forward, you have cells that join this subgraph because they differentiate into neurons. Or this subgraph, which is 26, which is at a slightly different time step. These are actually where the neural connectome starts to differentiate actually into different different modules. And so the undifferentiated cells will join these subgraphs over time. So that's why you have these lateral connections. So this is kind of a confusing diagram, but the idea is that as the embryo starts to differentiate and the number of cells starts to expand, you get these subgraphs. You can have exchange from like one generalized subgraph to a specialized subgraph as the cells differentiate, and then they're all essentially connected in some way, but we try to break them out into these lineages to show kind of the functional differences. And this this is what I call the density bifurcation model, which is basically a model that's similar to what they consider in graph theory as preferential attachment, but it's a little bit different than that because you can't use that model for a number of reasons. People do use it in looking at connectomes, but this is a different way of looking at it. And so, and then ultimately, we'd like to be able to model these types of embryos where you have processes going on in development. Not C. elegans, but like, you have Drosophila where you have some of these folding movements or other types of movements, spatially localized differentiation that's more distinct. They can really generate some of these subgraphs that really have different, they really have some interesting properties. So this one is based loosely on C. elegans. It's not, it's not exactly mapped to C. elegans, but it basically describes that process. In Drosophila, it would look far different than this. It would have a different sort of topology and how these trees branch, how the subgraphs look, and all of that. So,
Bradley?
Yeah.
Have you looked at the literature on trying to interfere with the development by killing an individual cell or by putting barriers between cells?
A little bit. I'm not really that familiar. I've seen it. Yeah.
There's literature which I dimly remember, going back quite a few years, where people would put a tiny sheet of mica between cells, presuming that represented a chemical barrier. Okay?
Yeah.
So the question is, given a hyper lattice like yours, can you interfere with it and predict what would happen?
Okay. Yeah. Yeah, that would be an interesting.
Yeah, I mean, maybe a search like embryo with mica might pull out some of that.
Yeah.
That would also be a physical barrier too. Definitely would change
Yeah,
the mechanics.
yeah. And nowadays, you can probably aim at a particular cell and destroy its nucleus, and then see if the cells on opposite sides make up for it or act differently than you expect.
Right. Yeah. Any other questions or comments? So we have this. I'm going to submit the abstract for this by March 11th, but I'm going to work on it.
There is a substantial literature on, I think, ultraviolet microbeam destruction of individual cells.
Yeah.
And if you focus through a microscope, if you focus a UV beam on a particular cell, you can destroy it, or even parts of cells.
Yeah. I think that's how they treat brain tumors. They use stuff like that to target the cells, and they're able to.
You use X-rays or gamma rays, or but there you're not observing what happens except grossly. You're trying to get into the whole tumor.
Yeah.
It'll be very interesting to compare, the altered network with what actually happens.
Right, the altered network. All right, so let me. I'm gonna move on to some papers that I have, and if you have to leave at 10, that's fine, or at the top of the hour, that's fine. But I'm gonna get into the. Let's see what we have here today. So Dick sent me this paper. This a couple days ago, I guess, and so this is called "The Time Complexity of Self-Assembly." And I'll zoom in here to the. So this is self-assembly. We've talked about active materials. We've talked the Ising models are sort of like in that category, although it's not quite. Anything like with Rule 30. Technically, that's sort of self-assembly, but the idea of self-assembly is where things kind of come together and form something on their own. And the idea is there's a physics behind that, and we don't really know how it works, but people have tried to figure it out. So this paper talks about the time complexity of this process. So this is a physical process. So we're talking about energy landscapes, and there's stat mech that's involved in this, just so we know where we are. So time efficiency of self-assembly is crucial for many biological processes. So there are a lot of biological processes. So when you get something that's where you get some self-assembly of molecules, for example, in development or even at the beginning of life, you get these self-assembly processes. Moreover, with the advances of nanotechnology, time efficiency in artificial self-assembly becomes even more important. So being able to do this is now an artificial system. And in nanotechnology, they're very interested in how do you assemble pieces of things into one sort of machine or something like that. So people are building machines at a very small scale. Sometimes it's at the nanometer scale. Sometimes that's a little bit bigger. But the idea is how do you direct this process? Because it's very hard to manufacture things at that process according to specifications. Furthermore, sometimes you don't know. In some systems, you just don't know how they spontaneously form. You can't really engineer that. So you have to kind of think about it as a self-assembly process. So while structural determinants in the final assembly yield are increasingly well understood, kinetic aspects concerning the time efficiency remain much more elusive. So this is dealing with kinetics, which is this energy and movement thing. So in computer science, the concept of time complexity is used to characterize the efficiency of an algorithm and describes how the algorithm's runtime depends on the size of the input data. So this is something that I actually got done with a paper with a collaborator where we talked about time complexity, and there's this big O notation that they use. Basically, it's this idea that your algorithm runs at has a certain runtime, or some problem requires a certain amount of search time to find a reasonable answer. So, like, there's some problems, if you've ever heard of NP-hard or NP-complete, those terms refer to the time complexity. And it's, usually non-polynomial, but there are other complexities that are much larger than non-polynomial, and there are problems that are much, much bigger than that. So our computers can solve things like maybe getting into NP-hard problems, but there are, NP-complete problems which are hard to solve, and then other problems which are even harder to solve with our computers. And if you think about like a self-assembly process, how do you overcome that time complexity with a natural process? So that's what they're trying to characterize here. Here they characterize the time complexity of non-equilibrium self-assembly processes by exploring how the time required to realize a certain substantial yield of a given target structure scales with its size. So this is their kind of where they're going with this. We identify distinct classes of assembly scenarios, or what they call algorithms. So they actually have designed these sort of, if you want to call them physical algorithms or what, but they're basically for self-assembly. What is the process to build this thing, and then what's the time complexity, or how long does it take to unfold? And then they show that this exhibits drastically different degrees of complexity. So as with algorithms, you see this complexity of different problem domains. Our analysis enables us to identify optimal control strategies for non-equilibrium self-assembly processes. Furthermore, we suggest an efficient irreversible scheme for the artificial self-assembly of nanostructures, which complements a state-of-the-art approach using reversible binding reactions and requires no fine-tuning of binding energies. So they're interested in this process of sort of trying to find ways to initiate self-assembly, but also how long it takes for different formulas to play out. So in nature, you see virus assembly, which is self-assembly. It must be fast to produce the many virus particles before the infected cell is eliminated by the host's immune system. So in this case, this is something that's evolved over millions of years, where viruses will invade a cell. The immune system will respond, but the virus needs to outwit that system, hit the replication machinery, and make more of itself. And so this is, something that is, of course, a process that has to be, it's optimized, of course, but it's also something that requires a lot of self-assembly within the cell. It's not just a matter of, 2 steps or something like that. So there are also larger complex nanostructures that we see in nature, but also that we want to realize for technology and medical applications. And so this time efficiency becomes vital. And so this is a figure 1 where they show this sort of scheme. So they have these different conditions where they look at, they have identical copies of S different species of monomer assembled into 1 or 2-dimensional or 3-dimensional structures with an edge length of L. So they're measuring the edge length and using that to look at the complexity of this thing that's assembled. Then they're taking these monomers and they're putting them together in different ways. And in doing so, they time everything and they see what the time complexity is. So some of these take a while to assemble. Some of these are very easy to assemble. And so then they're trying to characterize that. And so they have these different control parameters, dimerization, reversible binding, just in sequence activation. So there are all these different things you have to account for in this process. There's an activation or activation energy, there's attachment, and then you have this, you have a complete structure, an incomplete structure. So sometimes different algorithms will result in these incomplete structures which aren't really viable. Sometimes they result in these complete structures which are viable and they have a certain length. And so to build a structure at a certain length, how many steps do you have to go through? And then how long does it take? So we talked last week actually about like mutational hypercubes. And the idea there is like they basically take something like an RNA phenotype, which is mostly sequence. There's some secondary structure, but we're interested in the sequence. And the idea is if you mutate, so you can actually look at mutational steps to different changes or different phenotypes. And so then they build this network and say, if we start at point A, how many steps does it take to get to point B? And so you measure that out and you can tell how long, because you know how long what a mutation rate is, how long it takes to get from point A to point B. And it's a similar thing here where you have all these steps that you have to follow. And there are no shortcuts. Maybe there are shortcuts, but you want to characterize that. And so how long does it take? What's the time complexity and so forth? So this is interesting looking at it in this way. Now, I don't see any the conventional computer science notation for this. So I don't know what here's where they plot it out. So this is like complexity and time based on structure size. So in computer science, they would do like a big O notation. It would be based on some function, like either like a, something that's exponential or something that's, linear. Linear times are usually pretty slow. Exponential times are faster. So in this case, you have some of these scenarios where they map out the time based on the structure size, and they give you this idea of what it looks like. So that's basically what they're trying to do here. And they have a lot of different, they do a lot of different types of tests. So I think that's a very interesting paper. So yeah, the dimerization scenario turns out to be the most time-efficient scenario in all dimensions. Controlling the dimerization rate is efficient as it allows one to initiate just as many structures as needed, followed by a rapid growth phase if all particles are readily available. For linear structures, the supply control strategies rank second and third, the coordinated supply in the GIS scenario being more efficient than the uncoordinated supply in the activation scenario. Reversible binding is the least efficient approach to assembling large linear structures, but it is efficient for the assembly of higher dimensional structures, and then becomes competitive with the GIS scenario, slightly outperforming it for large structure sizes. So this is a little bit different than in computer science, where you have, your algorithms are basically just like, how you maybe abstract, the problem and how you represent it. In this case, you have these things that are actually physical things that you want to do, physical parameters, physical objects that you need to optimize. So there are different ways you can do this, and they link to different things in the system. So it's a little bit different than the computer science approach. And as such, you have these different types of scenarios, and they have, it's a little bit different than just like looking at an algorithm and saying, how do you optimize this? It's a lot more complex. So that's a good paper. Thank you for that paper. Let's see. The other thing I wanted to talk about today, last thing I want to talk about, is this paper on a fitness-based analysis of Daisyworld. So I don't know. I know we've talked about Daisyworld maybe in the distant past in this group, but the idea of Daisyworld is where you have this. It's a model of an ecological set of ecological regimes. So Daisyworld is where you have this planet, and the planet is filled with daisies, nothing but daisies, and that represents something that we see early in life, where you have like one or two species of microbe that kind of flourished and then took over the surface of the planet. So it's like it's like daisies of all one color, or daisies of a couple of different colors. So you have like a very small number of phenotypes that cover this planet. And so the idea is that you can run these dynamical simulations that show what happens when you have one color daisy versus two colors of daisies. The population proportion fluctuates over time, and you can actually see how what's growing on the planet can regulate the atmosphere of the planet. So you can measure different parameters of the planet's atmosphere and see how those different morphologies are affecting the atmosphere of the planet. And this has to do with the reflectance of the different colors and other properties of the daisies that are populating the planet. So it's a toy model. It's not really something that you would see in life, but it gives you some really nice insights. The problem with Daisyworld is it's not really linked to fitness. And so that's what they're doing in this paper, a fitness-based analysis of Daisyworld. So the Daisyworld model is sort of the basis for what they've called the Gaia hypothesis. So this is something that's, maybe about 50 years old or so. This was a hypothesis that life is regulating the atmosphere and the Earth functions as a self-regulating system. So what the Earth, what the biosphere is doing is regulating itself, and we can understand it through this model, but we can't really fully understand it because it's a toy model. So this never really sat comfortably within a mainstream biology, mainly because it didn't really appeal to a lot of the things that people were interested in, like fitness, like evolution, development. A lack of any clear role for evolution in the model has led to claims of teleology. So I know we've talked about teleology a bit. They talk about it in cybernetics, for example. There's a long history of that. But in biology, especially in evolutionary biology, teleology is kind of a dirty word because it harkens back to creationism and things like that, where you're basically saying that there has to be some external force that determines the flow of time. Of course, this is the sort of thing that when you come up with something, they're different fields and they have different sort of expectations for things. That's where that kind of comes into the clashes. So the idea of teleology here is that self-regulation emerges because it is preordained to do so. That's the criticism of this model. And this is, maybe more of an ecological model. But the idea is it's just kind of like hitting the idea of evolution just right, so that people are accusing this model of sort of promoting this idea of teleology. So that's the problem here. So the Daisyworld parable, a simple mathematical illustration of Gaia, went some way to addressing these critiques. So people have modeled this using dynamical systems theory and other things. It's agent-based models. You can build a nice agent-based model with this. But despite recent success in incorporating natural selection, and people have done this, it remains a widely held view that the ideas are inconsistent with biological principles. We show that a standard methodology for population genetics can be used to predict the stationary states and dynamic behavior of Daisyworlds. So this is interesting. So for dynamical systems, you can model this using dynamical systems. But the question is, well, what about the population genetics? And this is something that if you can model in terms of population genetics, it makes it much more biological, or it's rooted in biology more, because then you can say, well, there's clearly something going on at the genetic level that's being affected by environmental selection, and so we can do that.
You got to be careful. I think you can say it's rooted in population genetics. Whether population genetics is rooted in biology is a different question.
Yeah, that's true. But that's the idea. That's sort of that would be sort of a way to convince people. Now the question is what is that? How do you map from the population genetics up to the phenotypes? That's another question. So that would be another problem. Okay, so the system regulates its temperature due to the low-level evolutionary dynamics of competition. So between the thermally coupled daisies, and no higher principle is invoked. So basically, there are these daisies of different colors and in different states, and they're regulating the thermal conditions of the planet. And then that's selecting for certain daisies over others. and they're able to look at this in terms of, so population genetics, of course, is this idea that you have these different genetic variants. They have different frequencies in the population, and that those change with selection or with like processes like random processes and other types of things. So this is a model that we can see, at least, it doesn't just say that there's this sort of teleological change or that there's some other thing going on that we can't explain. This kind of roots it into what we kind of know as biology. So a reconciliation of Gaia with evolutionary theory may allow further development of evolutionary arguments for the existence of global self-regulatory systems. So this kind of goes through the idea of, this is James Lovelock who proposed this idea originally. And it's interesting because Lovelock was thinking in terms of life-induced feedback loops. So, this is a different sort of approach to looking at life. It's not necessarily not evolutionary. It's just that they're using a different way to look at it. So this basically shows something. It's just that you have to make a connection with the, different aspects of the organism more. And so only through stressing the unconscious nature of this regulation, particularly within reference to Daisyworld, has the Gaia hypothesis been able to refute claims of teleology. So people have done work on this to refute those claims, but the way to do this, of course, one way to build an effective model of Daisyworld and to demonstrate it is to build a spatial model where you have these daisies distributed in space. They have these different albedo types, which albedo is the reflectance off of a surface, and light surfaces reflect more than dark surfaces, which absorb heat. And so these have an effect on the overall condition of the atmosphere in this model. So, light-colored daisies will reflect heat back up into the atmosphere. Dark-colored daisies will absorb heat, and that has different effects on the model of the planet. And so it'll also affect the fitness of the daisies. But of course, there's no fitness imperative in the initial model. It's just basically the change of the phenotypes. And so people have worked on this. They've built different dynamical systems models. In this paper, they're building a model that's based on population genetics. And so it's it's kind of surprising that no one's done this before, because these are kind of easy to I would think they'd be fairly easy to relate, but this is something that people have kind of ignored. And so you basically have this model of the phenotypes. You have this model of population genetics, where you model a lot of these processes going on, and then you basically link them together. And so you end up with a different explanatory framework. Maybe it's more acceptable to biologists, maybe not. It depends on what kind of a biologist you are. If you're an ecologist, you probably don't care as much, perhaps. But this gives a concise, theoretically derived statement of the homeostatic nature of the Daisyworld system. Homeostatic in terms of fitness, not necessarily in terms of dynamical systems. So they're able, with a population genetics approach, they're able to give the phenotypes a fitness. They're able to link that to the genetics in some way. I mean, this is a toy model in and of itself, because you're simplifying the genetics of the daisies. You're basically saying that these phenotypes are generated by this genotype. And so but what you can do is you can create a fitness and look at the change in frequency of that phenotype through this model of the genetics. And so that's what they're trying to do here. And so you can also simulate this, and you can look at these simulation results. So now there's a probability density of the different phenotypes. You can see that they've been able to build that. They've been able to look at the regulation of temperature on the planet and the albedo. And so this is basically, it's a nice paper. It kind of goes through, it has some pretty decent mathematics in it. They do a lot of dynamical systems modeling and spatial cellular automata. So this is another paper where they do a lot of modeling, and it's a kind of a different approach to classical problem, as with our first paper. So we have Karan actually pointed out that they're one-dimensional cellular automata simulators. He put some links in the chat on that. And so you might want to check those out. Those are actually quite interesting. Daisyworld you can run as a cellular automata as well. I don't know if there are any models out there pre-made, but that's another option for making these kind of simple toy models of homeostatic regulation. Okay, so do we have any other questions or comments?
Do they get a mix of the phenotypes in the end?
I haven't really looked at the paper here. Let me share my screen. I'm not sure that they have like a table of what they've gotten.
In other words, is there a balance between the phenotypes, or does one dominate?
Right. All right. I don't know if they actually provide a good map of that.
No pictures of Daisyworld?
They may be later in the paper here. Let's see. Yeah, they don't really have any pictures of Daisyworld. They don't have any, like, snapshots of the simulation, but it's very mathematical, so they don't really give the... Yeah.
Yes. I'm sorry, seeing is believing.
Yeah, I know. It's... Yeah. So, yeah, they don't really talk too much about what those distributions look like. That's another thing about Daisyworld, is that it's one of these models that's sort of, the power of it is sort of the intuition. Like, you'll see the change in these different states, and you'll say, okay, I understand why, what this means, perhaps, or, what's going on, is that you have basically this change in state. Sometimes you can maintain multiple states, sometimes one state predominates. My guess is that you get a mix of those behaviors because, you see that in, like, population genetics. You get, like...
See, the next step is, the classical model of habitability of planets versus distance from their star?
Right.
Okay. Yeah. Can Daisyworld regulate itself to extend the habitable zone?
Yeah.
Might be an interesting question.
Yeah, it would. Yeah.
Okay.
All right. If there are no more questions, let's wrap it up for today. Thanks for attending. If you want to present anything, if you want to follow up on anything, please let me know or bring it to the meeting, and they can give you time to do it. All right. Thanks for attending. Talk to you later.
Okay, thanks. Bye.
All right, bye.